Hi, I'm Jack, and I'm in Ricardo Zayas' lab. And the question that we try to answer in our lab is, how do stem cells know what to become? And so stem cells are cells that have two particular characteristics. The first is that they can divide and create more of themselves, or self-renew. And the second is that they can differentiate to become other cell types. So maybe here I'll try to draw a neuron, and here maybe an epithelial cell. And what's critical about stem cells is that all the cells in your body, all the cells that comprise you, are either stem cells or their daughters. And so stem cells are very important early on in embryogenesis or in development. All the cells of the early embryo come from stem cells. But also in your body throughout your life, there's populations of stem cells that continuously divide to replace worn out or damaged tissues. So they're very important in understanding diseases of aging and in many cancers. And in some organisms, they can utilize these stem cells for impressive feats of regeneration. And so our lab uses the freshwater planarian, Schmidtea mediterranea, to understand this process. And we use this organism because it has the remarkable ability to completely regenerate from nearly any wound. So if we were to take this middle section, I'll draw it a little bigger here, and watch it over the course of about 2 weeks, what we would get back is a completely regenerated, fully functional planarian. A little bit smaller than the original, but after a couple feeds, it'll grow back to its original size. And this ability is based on stem cells, and it knows how to become its adult self again, because every cell within the planarian has within it, the DNA encoded instructions as to how to make the organism. But one thing to consider is that every cell within the planarian, indeed every cell within you, has the same set of DNA, it has the same set of blueprints. So how is it that the same set of instructions can be used to make different types of cells? And so part of that answer lies in the study of epigenetics, or how the DNA itself is physically packaged in the cell. And so in the nucleus, the DNA is wound around these proteins called histones. And how these histones are organized affects what parts of the DNA are accessible. So they can adopt this really open conformation like this. And in this case, these genes would be able to be read and be used by the organism to make that part of the blueprint. Or they can adopt this other conformation, which is much more condensed. And in this case, the genes would be off or inaccessible. And so this way, different parts of the DNA can be used in different ways. The question then is, well, how does it switch between these two? And part of that answer lies in that these histones have tails that stick out. And there's a class of factors that modify and affect these tails. And so we like to think of them as writers, readers, and erasers. The writers are responsible for putting on a mark. The readers can read these marks and switch between these states, and the erasers can get rid of these marks. And so in our lab, we study one particular complex. It's called polycomb repressive complex 1, or PRC1. And it works as a writer to put a mark onto a particular histone. It also works as a reader to recognize histones and to compact chromatin. And to understand the role of this complex in planarians, we inhibited certain subunits of the complex and observed what happened to the planarian. And what we found that was really interesting is that different subunits had different phenotypes. I mean, this was a little surprising because we think that this is a single complex and it works together, but the subunits have different functions, so maybe it's not entirely surprising. But that brought the question up for us, which genes are being regulated by the different subunits? And to understand this, we did what's called RNA-seq. So after inhibiting parts of PRC1, we looked at the total amount of messages in the cell that are being read out. And for one of the subunits we looked at, we found a lot of other epigenetic factors, which is kind of cool. And there's two ways that this could be happening. One, PRC1 could be regulating those epigenetic factors themselves, which are then regulating other genes that are causing the phenotype. Or something that could also be happening is if we inhibit PRC1, we're shifting this balance from this condensed state to this open state. And the cell might be trying to compensate by bringing other epigenetic factors into play to try to restore it to a balance. The second class, or the second factor, had a class of genes that are all related to the stress response. And this makes a lot of sense with the phenotype that we observed, which was a really mild regeneration phenotype. It was either delayed or inhibited. So the story that we think is happening here is that normally the action of PRC1 is to repress these stress genes in normal conditions. So if the cell's not stressed, they don't want the stress genes turned on. But when we get rid of PRC1 function, it's no longer able to repress those genes. Those genes come on and the cell thinks it's stressed. And that might help explain why the worms aren't as able to regenerate, because if the cells are stressed out, they're probably not as potentiated for regeneration. And this is all really important for us to understand because it does have impacts on human health, right? So it might be weird to think of a little planarian flatworm, but the fact is that humans have the same histone proteins and package their DNA in the exact same way. I mean, indeed, we even have the same PRC1 complex that has the same role. And so understanding in planarians what PRC1 is regulating can shed insight on what it might be regulating in humans. And also importantly, knowing what it's regulating in an organism that is regenerative, like a planarian, versus one that's not like a human can help us understand why planarians are able to regenerate. So thank you very much.